Thank you. Okay, so Rafi asked us to be clear to a general audience. So I hope that I will not be superficial, because I see that most of the lecturers didn't adhere to your directions. So let me start by the chemistry of the universe. It is very simple. The most important one. Hydrogen is the most abundant element in the universe. Oxygens appear by supernovas and so on and so forth, very rare. But this is with low redox potential. It has electrons to give, and this one wants to get electrons. And if you give it enough time, you have water. So essentially, the universe is very rich in hydrogen and very poor in oxygen. Okay? Now, if we look to our planet, we see oxygen all over it. It is with oxygen atmosphere. Not only this, because of the oxygen, the composition of the crust is totally changed. And it is absolutely unusual for the universe to have something that oxygen dominates the entire planet. As simple as that. So what caused Earth to be such oxidative reagent? It is oxygenic photosynthesis. And all of the oxygen that we have in the atmosphere and in the crust comes from oxygenic photosynthesis. Now, oxygenic photosynthesis probably started to operate about 3.5 billion years ago. And already at that time, after a few million years, there are evidences for the presence of cyanobacteria that are perfectly capable of doing oxygenic photosynthesis. Yet, for about 1 billion years, essentially, it was no oxygen in the atmosphere. The reason was that were metal elements, especially irons, that were oxidized in order to go down from the ocean to the bottom, and then to produce all the iron layers that now we dig for iron. And then the most disastrous thing happened, that oxygen started to accumulate, and most of the species, life species that lived in this point, disappeared from Earth. This was the most disastrous ecological thing that happened. And then you see that oxygen is not constant on Earth, and it depends on the photosynthetic activity. Probably this peak is around the Jurassic Park, that a lot of trees and everything was going very fast on Earth. Now, how oxygen came to Earth? It came from oxygenic photosynthesis that essentially is carried out by 4 huge complexes. Photosystem 2 that is capable of oxidizing water, and cytochrome B6F that shuttle electrons through the photosystem 1, and photosystem 1 that produce the most reducing agents in nature, and essentially it is responsible for the total energy in living systems on Earth. So we have two things that are unusual. One, that oxygenic photosynthesis take electrons from places that is unlikely to give electrons, water. And it gives electrons to something that doesn't like to get electrons. So it's pulling here, pushing here, and that is life, essentially. So there are many questions that we can ask about it. And let me pose the first one. Why do we need such huge complexes in order to carry the reactions? It makes no chemical sense. Because essentially, this is very simple reaction. Every color material can absorb light, and electron jump on. And this is the Einstein photochemical effect, a photoelectric effect. Quantum yield is 1. Now, in our systems, there are sometimes hundreds of chlorophylls and other pigments in which they absorb one photon. The photon can travel up to 150 places before it falls into a trap and it jumps. In photosystem 1, the quantum yield is very close to 1. Very close to 1. Now, a human cannot make 4 or 5 different pigments that absorb photon and will give electron with quantum yield of 1. So oxygenic photosynthesis is very unique in many properties, and we have to understand why it is so complex while doing very simple reaction. So to illustrate it, I will show you a distribution of pigment in one of the reaction centers, right here, and we'll see what happens if one photon is hit right here. So it will go that way, and it will go all the way from here to there. And it starts with quantum mechanical effects that we don't know now what they are. And it will go on through certain pathways, the excitation. And it will not go directly because there is no, in the structure, there is no place to go directly to the center. It will go around. Now, the explanation for this, why nature did it, is because the absorption of light is so fast that you have to stall time until you have biochemical reactions. It is as simple as that. But stalling time without losing quanta is really something that, of course, Jortner explained it for this part beautifully many, many years ago. But the entire thing is really, for me, still mystery, how you can do this kind of things. Now, in order to understand it, essentially, what we have to do is to have some structures. And there are many structures, and I will talk about the structures soon. But what the structures gave us, essentially, is a clear picture what happened in 3.5 billion years of evolution with the reaction center. So let me state to you that all the photosynthetic reaction center essentially came from one origin. It is homodimeric unit, something like this, that I was fortunate to discover with Ginter-Hauska many years ago in green bacteria, those bacteria that give the FMO, by the way. And then the first step in evolution, what happened, is gene duplication and heterodimerization. Now, this trick is really fantastic trick of evolution. Because the moment that you do gene duplication and you have heterodimer, it is, and you want now to complicate the system, what evolution is always doing, complicate structures all the time. It almost never make it simpler, only more complicated. So the moment that you have gene duplication and you have separate evolution of the two parts, and then when you want to add one more peptide, let's say here, it will not necessarily come to this site. And this site will be able to interact with another protein. So you can multiply the amount of proteins that you interact by a simple genetic trick. And it is extremely important, and it is going in almost everything in our body, this kind of genetic tricks. Now, from here, we have very nice pathway to photosystem 1 that is much bigger than what I painted here, and to photosystem. And essentially, this line is very interesting because this is the green bacteria that originally we discovered the Homo dimer there. So evolution leaves for us relics that really can guide us through the pathway of evolution for 3.5 billion years. And then from here, photosystem 2 was generated by splitting each one of these proteins into two, but in the middle, they are still totally homologous. Now this step, this is for photosynthetic bacteria that shed off all of the light harvesting that we have in photosystem 2 light, and it generated completely different light harvesting that I don't want to speak about it. Okay, so how do you go about crystallization of a photosystem 1 reaction center? So the story goes that way, that for my 60th birthday, I gave myself present. I wanted to become structural biologist. Now, every structural biologist starts with crystallizing lysozyme and solve it. I decided that the first protein that I will crystallize and solve it will be photosystem 1 from plants. And at that time, it was no single membrane protein that was published by X-ray crystallography from plants at all. No single membrane protein, no single transmembrane hélice. Photosystem 1, we knew already at that time, contained about 40 and something transmembrane helices. Moreover, it contained over 200 prosthetic groups. So you can imagine what kind of crazy person I should have been, but we call it in Hebrew chutzpah. And chutzpah, we have plenty of it in Israel. And essentially, this is the driving force for high technology and everything in Israel. So you take leaves, you prepare from them chloroplast, and then the biochemistry is extremely important. You have to clean it to this stage. And I took one genius student, Adam Ben Shem, and at no time we got big crystals like this. But it took him about 4 years to improve the diffraction of these crystals. And at the end of his PhD, he had no data to report on them unless he will solve the structure, and he solved it. And of course, then, this is Adam Ben Shem at that time. Now he's much bolder. And Adam came with the diffraction to this point, okay? 4.4 angstrom resolution. And with the help of excellent crystallographer that passed away a year and a half ago, Felix Frolov, we were able to solve the structure of photosystem 1, plant photosystem 1, at very poor resolution. And then a second student, very good student, excellent student, Alexei Amunz came. He came to 3.4 angstrom resolution. And this is Yuval Mazor. He came to about here. And now I will show you what happened right here. Okay, so Adam, and together with Felix and me, we published this Nature article in which we really solved the structure of plant photosystem 1 with poor resolution, but all the main ingredients were in the structure. So we saw the center core with new subunits that appear on the periphery, exactly like the evolution required. And for the first time, a second complex was attached to it, right here, that contained 4 different light-harvesting chlorophyll protein complexes. Okay? And correctly, we assigned each one of them according to the gene products that we had. That was a state of miracle because we saw very little. But you see that the chlorophylls are not defined. They look like biscuits and so on and so forth. And now we'll jump for the last few months. In order to jump for the last few months, we had to improve the resolution, but also we had to phase the structure extremely well. In a few months, we really invented something very nice. I won't go into it in the synchrotron. And we were able to get anomalous signals of phosphates and sulfates. And all of this gave us fantastic alignment of the structure that we have with the electron densities. And here, only for illustration, this is one of the chlorophylls that Adam showed in the structure. And this is the electron density of the chlorophyll. The electron density today looks exactly like this, of the same chlorophyll. So you see everything here, you can build the chlorophyll into it with no problem whatsoever. Moreover, photosystem 1 contains not only chlorophyll A, but chlorophyll B. The only difference is one oxygen atom right here. We see the oxygen atom extremely clearly at this 2.3 angstrom resolution. And we see the different carotenoids with these electron densities that there are no doubt that this is beta-carotene, lutein, zeaxanthin, and xanthophyll. So we see all the important ingredients, all the amino acids that we have in the structure. And the structure looks something like this, which for X-ray crystallography, it is just about the limit. There are 16 subunits, 45 transmembrane helices, 135 prosthetic groups that are not proteins. All of them are there, and here is the list. And for the photochemistry, it is important to note that the chlorophyll concentration is 0.46 molar, and the carotenoid concentration is 0.11 molar in the structure. So nature did something marvelous. We cannot mimic it in any case. Okay? So now we look at the pigments in the structure. So green is chlorophyll A. But you see that chlorophyll B is also present and not at the same distribution in all the four chlorophyll protein complexes right here. And the carotenoids are in blue. I don't specify which one of them. As I said, each one of these pigments, when it absorbs photon, excitation will happen, and the excitation transfer will go through this place. Now, according to the structure, there are two main pathways for the excitation transfer, from here and from here. And lo and behold, look what's going on here. A chlorophyll B stuck right in the excitation transfer, right here and right there. And chlorophyll B has higher energy. So the only way to conciliate with this is, and the chlorophyll B was put in nature, is there for a purpose. So the only way to conciliate with this is to consider few chlorophylls together as a quantum mechanical entity that use also the protein vibrations in order to pass here easily from one entity to the other. Okay. So essentially, I described to you the end product of this branch of evolution. And I will mention this one later on. But in cyanobacteria, most of the cyanobacteria has a trimer like this. And the first photosynthetic reaction center from oxygenic photosynthesis that was crystallized in Berlin by Horace Witt and his colleagues, Petra Fromme and others. And this was a tetramer like this, exactly like this, from thermophilic cyanobacteria. And they decided in a meeting in Berlin that in mesophilic reaction center, they will not be able to get good resolution. They solved it to 2.5 angstrom resolution. Now, what they did, essentially, they refined only one of the trimers and multiplied by 3. And because at that time, in 2001, the technology and the synchrotrons were not as good as we have it now. So I will show you our structure of mesophilic reaction center from synicocystis at 2.5 angstrom resolution. But we refined the entire megadalton altogether. And when we refined the entire megadalton altogether, we got surprise. It is a trimer of the same gene products exactly, but they are not identical at all. And you can see the distribution of the different entities here in inside, and we see many, many of them. So let's look only on the carotenoids. So you see that this is the distribution of the different carotenoids in the trimer. And you see that none of the carotenoid numbers are divided by 3. So each one of the monomers have different carotenoids. And the most peculiar one is this one, that is only one of these carotenoids. So at high resolution, we really can detect a lot of things that we were not able to see before. Now, I won't go into it, but I would like to state that evolution in general goes from symmetry to asymmetry. And in chemistry, we like to have symmetry all the time. Now, we will have to close the gap between the symmetry and asymmetry, and this will be for the future that you ask us. I'll give you another one later. Okay. So, another interesting thing is that we have this homodimer, heterodimer, and this is eukaryotic reaction center, and this is prokaryotic reaction center. They branched out about 1.5 billion years ago. And one of the relicts of this kind of branching is red alga. And the red alga photosystem 1 reaction center is probably the most ancient one from the eukaryotes that we know now on Earth. So very recently, last year, we published the structure of this photosystem 1 reaction center. I won't go into it, but it really a hint what happened on the way in the beginning from here to here. And this is the structure that we got. So we got it to a resolution that is not very high, but the PDB is right here. The same day that we published this PDB was published from our X-ray crystallography, cryo-EM structure also was published from the same green alga. So cryo-EM takes over big complexes, there is no doubt. Unfortunately, we don't have cryo-EM instrument in Israel. And this is one of our failures that we don't have it. Because now we are lagging behind 5 years. And if we are lagging behind 5 years, to catch up with the entire world, it will take many more years. and we will only scratch their tails when we come out. So what we are doing, Ada Yonat and me, collaborate with Alexei Amunds in Sweden, and Ada already published her paper with him, and I hope to publish my paper. But this is not the way to go about it. Israel must have cryo-EM instrument. In Beijing, they have over 10 instruments like this. Okay, so this is for the political person. Okay, so in the last few minutes, I want to tell you a story that I'm not sure that many of you are aware of it. We look once again at our planet, beautiful planet. Now, what we don't see in the ocean are viruses. And believe it or not, viruses in the ocean contain more genetic information, tenfold more genetic information than all the organisms together, from bacteria to whales. There is an enormous amount of bacteria, phages in the ocean, viruses. And what these viruses are doing, they are doing us a big favor because they control the population of cyanobacteria and green algae such that they will not be able to dominate the oceans. Because if they will, one of the species will dominate the ocean, it will turn into a stinking pond. Okay? So every time that you have bloom of this cyanobacteria and green algae, the viruses attack them and shut them down, and the bloom will come 10 years later, and the cycle will come again and again. So they also do big favor for us. But a few years ago, Oded Beja in the Technion discovered pieces of photosystem 1 in one of the marine viruses. He came to my laboratory, we collaborated, and we discovered an operon that is encoded by marine viruses. And this operon now is present in about 100 different marine viruses. And this is amazing. So the biological story is not quite clear. So what we are doing biochemistry and structural biology. We do mimetic viruses. So we do what the virus wants to do, but we plant it into cyanobacteria, isolate it, study it, and I will not have time to go over it, crystallize it, and solve the structure of the mimetic viruses. And it is promiscuous. I don't want to take the time for this. So if you compare an important point right here, that this is where the electrons are entering photosystem 1 by plasmocyanin, you see the main differences between the virus-encoded one and the cyanobacterial-encoded one. So what in all the viruses happened is that here, there are two subunits, PSAJ and PSAF. And PSAF, because it starts with the N-terminus right here, it needs a leader sequence in order to stick it to the other side of the membrane. What the viruses did, something very clever. They had a fusion protein between PSAJ and PSAF. And they don't need leader sequence because PSAJ leads the entire structure, and it is provided. At the same time, what the viruses eliminated the specific binding for plasmocyanin that is present in the wild type, and here it is not there. And they became promiscuous to electron donors. So if you have a trouble, you want to be promiscuous to take electrons from whoever will give you, and this is what the viruses are doing. Then we asked ourselves, why we need such huge structure like trimer like this in syndicocystis? So we started to eliminate subunits and pieces of photosystem 1, and we came up with minimal photosystem 1, still can support oxygenic photosynthesis in the cyanobacteria. It's slower a little bit. But you see, as an example, that nature could settle for something like this, and they end up with this. So biology and evolution complicate and extend the complication all the time. It takes it out of symmetry. It makes bigger and bigger complexes, and there are other organisms that the complexes are even bigger. Okay, so I want to end up with something that should consider here blasphemy. To personalize or make the second law of thermodynamic entity, not a law. Okay? So entity or person can punish you for something wrong that you are doing. And this is exactly what I want from the second law of thermodynamics. So the future of humanity is not very bright because we dig in every year, we burn what photosynthesis buried in the ground in 1 million years. Now, I'm not talking about warming, oceans, nothing. Thermodynamics doesn't allow you to do abrupt deviation from equilibrium. They like very much to go with equilibrium and change it to better and better and better, but not to do abrupt change. And this is really abrupt change that it will cost. So essentially, we have to stop doing it and to look for other sources for energy. And I'm certain that utilizing oxygenic photosynthesis in a sensible way will be part of the solution. However, second law of thermodynamics also suggests that excessiveness should be forbidden. And excessiveness, and I will sound like an old guy. I think that the fact that some people now build rockets to send two rich people out to the outer atmosphere to look at the beautiful Earth and waste energy, what photosynthesis buried in 1,000 years for this should be forbidden. It's as simple as that. And you can go for excessiveness in money and so on and so forth. So the second law of thermodynamics will rule much better than our government. And I would like to thank, of course, the sources of money. ISF, this is right here, supported me all the time, and I'm very grateful for this. I was extremely lucky to get ERC, the first in our faculty. It is gone by now, but that's okay. ICOR, that's another thing that is also originating from here. And I would like to mention the synchrotrons in Europe. Synchrotrons in Europe are fantastic institutions. You won't believe that they are situated in Europe. No bureaucracy, nothing. You go, you do your measurement. Everybody helps you. Try to do the best. You give good proposal, they give you time as much as you need. And I used 3 of them, BESI for us in Berlin, ESRF in Grenoble, and SLS in Switzerland, and I'm very grateful to them. Thank you.
Thank you very much. I have to say, I'm not qualified to comment on the politics, but the structural biology is awesome. Thank you.
Thank you.
Questions to Professor Nelson? Yes, please.
Thank you very much. Very interesting. you showed the two chlorophyll B molecules, and they looked quite far apart from the reaction center.
Yeah.
Why do you say they block the energy transfer?
No, they block the excitation transfer from the LHC moiety, as I showed it. The excitation transfer goes through this pathway.
Okay, thank you.
Yes, please. Igor?
So you mentioned that when you started to crystallize this protein, there were no structure of plant membrane proteins.
That's true.
Is there something intrinsic that makes it more difficult to express
no,
membrane proteins?
Membrane proteins were extremely rare. For the bacterial reaction center, 3 people got a Nobel Prize, and it was not that far away. So membrane proteins were rare. So the
I mean specifically plants versus bacteria.
Plants, I don't think so. If your biochemistry is good, you can crystallize it. It's nothing special. The funding is less for plants, so it came later. Okay.
With your permission, I'd like to speculate a little bit about your comments about asymmetry and why there is asymmetry. And this may be a prelude to Greg's talk, and I was talking with Greg about this yesterday. So, of course, the wavelength of light is much larger than the size of the molecule. That's the dipole approximation. So, in fact, whether you want to consider quantum mechanically or not, the light absorption is distributed. And so, at least from this point of view, the excitation is also distributed. These are all highly coupled chromophores.
Yeah.
And so, how then does the distributed absorption of light become a localized concentration of energy? And so, it could be that the asymmetry plays a role in this localization, that the breaking of the couplings between all these coupled chromophores may be helped by the asymmetry of the problem.
No, I'm sorry to not to agree, because I saw the carotenoids. When carotenoid absorb photon, at no time it goes to the neighboring chlorophyll. So, and it is gone. It cannot go back. So this cannot be the reason for the carotenoids to distribute differently in the different units. But
I'm not saying that the carotenoids absorb light. I'm saying that the carotenoids are involved in the decoherence of the coherent absorption of light across the chromophore.
Well, yes. If this is the case, I accept it. Yeah. Yeah.
There is one dilemma prevailing in all these systems, and this is the symmetry breaking.
Yeah.
One is one branch of the reaction center effective amount.
Yeah.
How universal it is, and what progress did we make during the last 20 years?
Okay, so in photosystem 2, only one branch is productive anyway, because the quinon accepts only from one of the branches. In photosystem 1, the 2 branches are productive, but not equally. And people try to distort it and to measure it, and they come with different numbers, from 10% to one of them up to 50-50. And we don't understand it yet. But I think that I have, for biochemists, the tools are very important for all the biologists. And I have now another reaction center that we solve the structure from alga, in which I will be able to eliminate one of the quinons. And then I will be able to look for it. Okay, because I know that there is empty space there that I can probably pull it out.
Just a minor technical question. You showed the structure, over 870, if I noted correctly, water molecules.
Yes.
My question is,
yes.
Are all these water molecules kind of loose hydration water, or are they fixed that you see them?
Okay, so there are different groups, and I didn't have time to dwell on it. This is also fascinating. 7 of the water molecules are exactly at the same position as in the cyanobacteria. It means that these 7 water molecules are there for 3.5 billion years, and nobody removed them. Okay? So, thank you for the question.